My study is a phase 3 trial that was designed to determine whether targeted therapy is superior to chemotherapy in older patients with previously untreated chronic lymphocytic leukemia, or CLL. Prior to this study, most patients who were over the age of 65 were initially treated for CLL with a combination of bendamustine plus rituximab, which is a chemotherapy regimen that's very effective, but also does have significant side effects for many patients. We sought to determine whether a targeted therapy called ibrutinib, which is an inhibitor of Bruton's tyrosine kinase, or BTK, was superior to bendamustine plus rituximab when it was given either alone or in combination with rituximab. After our study was completed, we did find indeed that targeted therapy either alone or with rituximab was superior to bendamustine plus rituximab. At 2 years of follow-up, 74% of patients treated with chemotherapy were progression-free, while 87 and 88% of patients treated with targeted therapy were progression-free. This study therefore changed the standard of care for patients with untreated chronic lymphocytic leukemia.
The project that I'm going to tell you about involves a breakthrough, life-saving discovery and development of an innovative precision-based therapy in a 12-year-old child with a complex lymphatic disorder. The project began by our discovery of a new mutation in a gene which is called ARAV, or RAV kinase, and this opened up a completely novel pathway for this complex, life-threatening lymphatic disorder. The mutation was a gain-of-function mutation, so the pathway was on all the time, and this drove a proliferation of cells that resulted in fluid leakage into the heart, lungs, belly, and extremities. We subsequently took the mutation and we put it into a zebrafish, and the fish developed a lymphatic phenotype that we saw that we could rescue by giving an inhibitor drug, which was an adult cancer drug only approved for melanoma cancer. We subsequently took that same medicine and we treated cells from the patients, and we saw that we could contain the hyperproliferation or the overreactive state of the cell growth in the patient cells. We then went and asked for compassionate use single IND approval from the FDA and the IRB, and it was granted, and we started treating the patient with an inhibitor against this particular overreactive pathway, which is called the ERK signaling pathway, with a medicine called a MEK inhibitor. And the patient responded very rapidly, and in a few months resolved more or less all of his symptoms. Lung function went back to normal, and he is currently involved in daily activities without hardly any compromise at all, including running track. He couldn't stand up from a chair because he was so swollen before. We've since treated 7 patients, all of whom have mutations in different genes in this pathway, and they have all responded in a very comparable, magnificent way.
The Credence trial was a trial that was designed to study canagliflozin, which is a drug that people with diabetes take to lower the blood sugar. And we wanted to evaluate if canagliflozin improved kidney function in people who have type 2 diabetes and have kidney disease with it. Credence showed that canagliflozin reduced the progression to kidney failure by 30% compared to placebo. Importantly, it also reduced cardiac death and hospitalizations for heart failure by 31%. This finding is really, really important because it is the first drug in 20 years that shows that you can improve kidney outcomes and the progression to renal failure or kidney failure and hemodialysis in people with type 2 diabetes.
Multiple myeloma is basically very resistant to most therapies. Although we have treatments now that can keep patients alive for up to 5 or 6 years, eventually the multiple myeloma becomes resistant to therapy and the patients almost all die of multiple myeloma. We decided to treat the multiple myeloma with a T-cell therapy. We remove the patient's own T-cells from their body, genetically engineer those T-cells to express a receptor that gives the patient's own T-cells the ability to recognize and kill the multiple myeloma. This treatment, invented first at the NIH, in my lab and then went on to test first clinically here at the NIH. Our first results were quite promising. We saw an incredible decrease in multiple myeloma in many patients. This led to a multi-center trial in collaboration with industry partners as well as other extramural collaborators in many universities across the country, where we tested this in advanced multiple myeloma patients with very few other treatment options. We had an 85% response rate and an 11.8-month progression-free survival rate. Phase 3 trials are actually very far advanced at this point. Many patients have been treated on the phase 3 trials. Results have continued to come in that are promising, and this treatment is hopefully progressing to a commercially available product so it can be used to help more patients in the future.
Multiple sclerosis affects people in the height of their life or careers and destroys their mind and ability to do things. Traditionally, there's no cure, and you're looking at only trying to slow the rate of progression of the disease, the rate of damage to the central nervous system. What this research has done is shown in a randomized trial. It not only slowed progression, but it actually improved neurologic disability, and patients, the vast majority, went into very long-term remission without new evidence of disease activity for over 5 years. It's taken a chronic disease and with a one-time treatment turned it into an acute illness that you can recover from. The most rewarding thing is to see them come back and that they're better, because when you're healthy, you take it all for granted. The patients are so grateful and so happy, just glad to have and to appreciate what you had and lost and thought it would never come back.
The main question we were trying to answer is whether or not Smartwatch technology can be used to identify atrial fibrillation. We enrolled over 419,000 people in a course of 8 months, so one of the largest cardiovascular studies ever done. The Smartwatch was able to monitor for an irregular pulse, and if they qualified, they were mailed a patch monitor that they were able to apply on themselves. After a 7-day period of wearing this patch monitor, they simply had to mail it back. We were very excited to see that only a very small number of these participants had an irregular pulse notification. And as we later saw, many of them were having atrial fibrillation when they had that irregular pulse. This study is important because it really serves as a model for future studies that would like to perform clinical trials in a completely virtual manner. From the very beginning to the completion of the study, it can be done without participants having to physically go to a hospital or a clinic.
Over the 60-year period that hypertrophic cardiomyopathy has been a clinical disease, sudden cardiac death has been perhaps the most visible and certainly devastating complication of the disease. And we live, of course, now in an era where contemporary medical therapy, in this regard, the implantable cardiodefibrillator, or ICD, is available to treat life-threatening monomorphic VT tachyarrhythmias and to prevent sudden death. And this has put an enormous emphasis, therefore, on identifying reliably those patients that are most at risk for sudden death events, who would be the best candidates for primary prevention ICD therapy. Our study evaluated the efficacy, really, of a risk factor algorithm that we have applied, which was based on a number of individual major risk markers that have been previously established and are part of ACC/AHA guidelines in 2011, as well as a number of new, novel high-risk markers as well that have emerged over the last decade. For patients that had one or more of these major individual risk markers, we considered them to be at a risk for sudden death that was increased enough that it was reasonable that they pursue primary prevention sudden death therapy in the form of the ICD. And what we found over our follow-up period was that there were 82 patients who received an appropriate shock for potentially life-threatening ventricular arrhythmias that were implanted because they had one or more of those major ACC/AHA risk factors.
For parents who've had a premature baby, all vital signs of that baby need to be monitored. And the way that happens currently is with a rat's nest collection of wires and adhesive tapes that bond sensors to the surface of the skin. Our goal was to reproduce all of the kind of functionality that's currently provided with these wire-based sensors with these wireless battery-free skin-like devices. Implementing the antenna systems and the data recording hardware that go along with these devices and deploy them in a real operating NICU. It softly interfaces with the skin and allows us to measure electrical activity associated with beating of the heart, so we can capture EKG waveforms. What we're able to do now is correlate changes in heart rate, changes in temperature, changes in blood oxygenation that occur as a consequence of the mother holding the baby. And so our goal here is to make the wires go away and enable the parents to interact more naturally, more frequently.
Peanut allergy is a very common reason for life-threatening allergic reactions that land people in the emergency room. And peanut oral immunotherapy is an approach that's been studied to treat peanut allergy. The POISE study looked at 120 peanut allergic children and adults and randomized them to receive peanut oral immunotherapy or placebo over the course of 3 years. 85% of those who received peanut oral immunotherapy were able to tolerate 16 peanuts without an allergic reaction by the end of 2 years, compared to 4% in the placebo arm. Safety measures continued to improve the longer you were on peanut oral immunotherapy, and reactions to accidental ingestions from peanut decreased the longer you were on peanut oral immunotherapy. And what was really exciting is for the first time we saw that blood tests done before patients started therapy were able to predict successful outcomes at the end of therapy.
For many years, our nation has invested a large amount of money in trying to discover ways to prevent and treat dementia. And so this study, Sprint Mind, provided the first evidence in history that we can actually do something in the physician's office to reduce a person's risk for developing dementia in the latter years of their life. 5 years ago, we already established that a blood pressure goal of less than 120 millimeters of mercury, systolic blood pressure, actually reduced heart disease, heart failure, stroke, and heart death by 30 to 40%. Now we've shown that you can reduce memory loss, early dementia by 15 to 20%, doing the same thing. More than 80% of people over the age of 85 and more than 50% of people over the age of 50 have elevated blood pressure. And so Sprint Mind showed a clear relationship between blood pressure control and reducing the risk for dementia, which is a major public health goal for the United States. For years we've been saying, do what's good for your heart. Now we can say that what's good for your heart in terms of lowering blood pressure is also good for your brain. And that was not known until this year when the study was published.